Okay, I'm going to start things off by talking a bit about a few things about preparing DNA for HiFi sequencing. Then I'm going to discuss our different HiFi library prep protocols and the considerations that need to be made for each one. After that, I'm going to highlight a troubleshooting case study that I hope is helpful for everybody. And then I'm going to finish things off by going over some frequently asked questions and then taking questions from today's audience. Okay, so when planning your HiFi sequencing project, there are three key attributes your sample should have in order to maximize your yield. The first is to start with freshly extracted DNA from fresh or flash frozen tissue. And once extracted, you want to proceed to library prep in a timely manner. For example, you want to proceed within a couple of weeks of extracting DNA rather than waiting, months or even years. This is because DNA will accumulate damage over time just from hydrolysis. This is not to say that old samples cannot be sequenced. It's just that using fresh DNA will help maximize that yield and avoid any possible issues with damage that can reduce your read lengths. The second attribute is purity. DNA should be free of RNA, should be free of polysaccharides, any inhibitors, dyes, and salts. It should just be DNA. And then, finally, you want to ensure that the DNA is of high quality. For PacBio HiFi sequencing, you no longer need, super high molecular weight DNA. You don't need things of hundreds of kilobases in length or megabases, but you just want it to be, pretty good quality with a minimal amount of degraded material. So generally, we say now, the majority of your fragments should be greater than 20 kb in size for most of our HiFi sequencing protocols. The only sort of exception to this rule would be with metagenomic samples. Oftentimes with those kind of preps, extracting DNA from metagenomic samples, they're going to be highly degraded. What's important there is that they're pure. So with those kind of samples, just ensure that they're pure. You don't need super high weight DNA to do any HiFi sequencing. We have a tech note that lists a dozen different commercially available DNA extraction kits. Other commercial kits or methods may work fine. These are just the kits that we know firsthand from our customers that have produced high quality DNA and good HiFi sequencing results. Now, sometimes commercial kits make the proteinase K and RNase A treatment steps optional, but I highly recommend that you include these steps even when extracting from easy to lyse samples like whole blood or frozen cell pellets. It's important to get rid of all those, any possible nucleases or proteins in there. If you're studying an organism where a commercial kit might not be appropriate or just is not going to work, we have a webpage resource that Jonas Korlock, our CSO, put together. It's called extractdna for PacBio.com, and it lists publications by the scientific community describing protocols for extracting high molecular weight DNA. The resource is sort of organized phylogenetically, so this just is to help you find a protocol, similar to the organism that you're working on. So yeah, highly recommend checking this out if there's not a commercial kit solution for you. Okay, so once you have your DNA, one valuable QC method that I frequently employ is to quantify my sample on both the nanodrop and qubit systems. A pure high molecular weight DNA sample should exhibit relative agreement between the two systems, despite the fact that the nanodrop is measuring absorbance from all nucleic acids. If there is a large difference between readings, for example, greater than 50% of the qubit reading, I recommend doing a round of Ampere PBB purification. Then after that first round, go back, measure the concentration again on both the nanodrop and qubit systems, and see if the difference in concentration is reduced. They also recommend checking to see if there's an improvement in the 280 over 260 or 260 over 230 ratios. If you see an improvement, I would continue with additional rounds of bead purification until the concentrations are in relative agreement with each other, and those absorbance ratios are within our recommended range. Now, Ampere PBBs cannot rid a sample of everything. So if you don't see any improvement in your sample after that first round or multiple rounds, then I'd consider stopping and thinking about another approach. Now, one of those other approaches might be trying the QIAGEN Power Clean Pro Kit. Our customers have reported using this kit successfully for cleaning up their samples. I personally haven't used it, but I think this should work. However, there's a couple things you should watch out for. The most important being that QIAGEN recommends that the DNA fragment sizes need to be in the range of 60 base pairs to 50 kilobases. So it's probably best to use this on sheared or slightly degraded samples. The second important thing to kind of look out for is to expect a lot of sample loss in the whole process of the cleanup process here. So it's probably best to kind of go in with twice as much DNA than what you're actually going to need for library prep. Alternatively, if you don't want to take those kind of risks, some people have reported that just doing another round of the standard isopropanol precipitation and the 2 rounds of just washing the pellet with 70% ethanol and resuspending it in a lution buffer will also work. So these are some alternatives to doing the ampere bead purification. So let's move on and talk about the differences between our HiFi sequencing protocols. So currently we have 3 HiFi sequencing protocols that are used for Genova Assembly and/or variant detection projects. I'm going to start by going briefly over the workflow of each protocol to sort of highlight the differences. But then we're going to go in a little deeper and talk about genome size and how that's going to influence DNA input amounts. So I hope most of you are somewhat familiar with our standard HiFi sequencing protocol. This protocol, states you should probably start with 3 to 10 micrograms of high quality genomic DNA, depending on the genome size of the organism you're working with. You'll then shear that genomic DNA down to average size somewhere between 15 and 20 kb. You're going to take that shared DNA and bring it into our SMRTbell library prep construction. And then you're going to finish things with a size selection step, either using the Sage ELF, Blue Pippin, or Pippin HT platforms. Once you have your size selected library, then you're going to run it on our Sequel IIe system using the standard 30-hour movie. The low DNA input protocol is very similar to the standard protocol. You're still going to share your DNA and then use our SMRTbell library construction protocol. The big difference here is that you're going to use ampere PP beads to do the size selection step. And this helps minimize sample loss, but you only can remove fragments that are less than 3 kb. So because of this limitation, it's really critical to start with high quality genomic DNA. So we say ideally, when you're first QCing your sample, you want that average mass to be greater than 30 kb. But it's really important here. You just want to have a sample that has a minimum amount of degraded material, especially less than 10 kb, because it'll be very hard to remove that material, and then that material is going to impact the fragments that get sequenced, so you'll have a lower average read length. So similarly for our ultra low input protocol, it's critical to use DNA that has a minimum amount of degraded material. And the reason here is that, after shearing, so we're going to shear that genomic DNA down to average of 10 kb. So once we have that, we're going to ligate linear adapters to it. And now those linear adapters are going to be used to amplify those fragments up. So if you have the presence of a lot of shorter fragments, those are going to preferentially amplify. And it'll be very difficult to nearly impossible to remove them, even with a Blue Pippin step, because you just won't be generating enough of the 10 kb fragments. So again, yeah, it's really important to start with good quality material here. Okay, so which of these protocols you use is going to be kind of determined by the genome size of your organism, the amount of genomic DNA you have or can get, and the molecular weight of that genomic DNA. So first, let's talk a bit about how genome size factors in to each protocol. So currently with the Sequel IIe system, using our sequencing chemistry 2.0, you can expect to get up to 35 gigabases of HiFi yield per smart cell when using inserts between 15 to 20 kb in average size. So what this means is that for 1 gig genome, all you need is a single smart cell to reach at least 25x coverage. I chose 25x coverage here because from a lot of our customers that work on Genova Assembly, this seems to be the point at which contiguity and completeness of the assembly begins to level off with HiFi reads. This is also true if you're looking at variant detection. There seems to be the sensitivity, specificity, or sort of plateau around there as well. And this all scales up linearly. So for a 2 gig genome, you're going to need 2 smart cells to reach that type of coverage. And for a 3 gig genome like a human, you're going to need 3 smart cells to reach that 25-fold coverage. So the number of smart cells required for your project will then determine sort of that minimum input amount of genomic DNA. So for example, what I've shown here on the table on the left-hand side of the screen is the minimum amount of SmartBell library needed for sample setup using a 50 picomolar on-plate loading concentration. Now, for those of you that are new to our technology, this is the step where we bind the sequencing polymerase to the DNA library. So shown here, at minimum, for inserts between 10 and 20 kb, you need 75 to 150 nanograms of SmartBell library per cell for the Sequel II system. To calculate how much genomic DNA that is, we can back calculate, assuming a 10 to 20% total yield from genomic DNA to the final size selected library using our standard HiFi protocol. Now, for the low input and ultra low input protocols, this yield number will differ dramatically because of differences in the protocols that we just discussed. But what this does show is that at minimum, all you need is 4.5 to 5 micrograms of genomic DNA to get a complete human de novo assembly with 25x coverage using HiFi reads. So that often surprises some people because it's a lower input amount than what I think is even stated in the HiFi protocol. So it's really good to be aware of that. And the final consideration that needs to be factored in is DNA quality. So shown here is the femtopulse analysis of 4 individual mosquito genomic DNA samples. So samples 1 and 2 look great. You can use these with any of the 3 protocols, the standard, the low input, and the ultra low input. But samples 3 and 4 just don't have enough high molecular weight DNA to proceed with the low or ultra low protocol successfully because of limitations around size selection there. It might be possible with enough material for a sample like number 3 there to use our standard HiFi protocol because it's going to include a size selection step, right? You're going to get rid of a lot of that lower material, but you'll just need a couple micrograms of material going in to make that possible. Another important aspect here too is if you are working with a small organism and plan on using like the low DNA input protocol or the ultra low DNA input protocols, I think it's important to plan on extracting from multiple individuals like we show here, because with small organisms like that, there is going to be variability in the quality of DNA that comes out of that extraction process. So plan on, getting DNA maybe from 5, 6, or even like 10 individuals, and then QCing them and picking the best extraction to proceed with. Okay, now that we covered, the main considerations that need to be factored in when planning your HiFi sequencing project, I wanted to talk about troubleshooting using a real sample that I prepared around this time last year. The sample I chose to highlight is the maize line B73. So we received this DNA from a collaborator. And the first thing I did was, it was pretty viscous to begin with. So I mixed the sample using a P200 wide board tip that kind of better homogenized the sample. And then I made a small 1 to 10 dilution of that sample. I then, took the concentration readings on the nanodrop and qubit systems. And now what I noticed right away was those concentration readings were drastically different, as I'm highlighting here. So just kind of going on a hunch, I checked to see if there was RNA present using the RNA broad range assay from on the qubit system. And sure enough, the qubit system indicated that there was a high amount of RNA present in the sample. So then I proceeded with just doing a standard 15-minute RNase A treatment at 37 degrees. And then I cleaned that reaction up just using Ampere PB beads. And then after doing this, I took the concentration readings again, both on the nanodrop and qubit systems. And this time they were much more in agreement. In fact, only differed by 13 nanograms per microliter. So this was really good to see and indicated that the RNase treatment was working. Not shown, but I did do a second RNA broad range assay reading on it, and it confirmed that there was no longer any RNA in the sample. Okay, so once I confirmed that the sample was clean, I proceeded to sharing it on the Genome Megaruptor 3 system using a speed of 31. So this gave us a good mode peak size. So I took 5 micrograms of that shared genomic DNA into our standard Express template prep kit 2.0 library prep protocol. When I got to the size selection step, I decided to experiment a little bit using the Sage ELF 10 to 40 protocol. In order to get size fractions greater than 20 kb, we kind of wanted to see how the assemblies would differ between sequencing a fraction that was greater than 20 kb versus one, say, like that was more like around 15 kb. However, the size selected material didn't sequence well, and then we later discovered that this particular protocol was likely the culprit. Therefore, I really don't recommend using it, but if you do want to isolate sort of larger fractions using the Sage ELF system, then you can contact Sage Science. I think they have some custom protocols you can use to kind of work around this. But for most applications, our standard 1 to 18 kb protocol that's in the standard HiFi protocol should work fine for your purposes. So, because that sample did not sequence well, which I'll show you towards the end of the presentation, we need to get more DNA. So we had let the collaborator know about the issue with the RNA when we ordered a new isolation. Despite this, the new isolation still contained RNA, still contained some RNA. So I proceeded again with the RNase treatment using the Ampere PBB cleanup and the standard shearing on the Megaruptor 3 system as I did previously. This time, though, we decided to use the blue Pippin system for size selection. I split approximately 5 micrograms of the library between the two lanes and just used the protocol that's described in the standard HiFi protocol. So this is doing 2 fractions on the blue Pippin. First, you isolate a 9 to 13 kb fraction, and then you manually elute everything above 13 kb. So I took the above 13 kb fraction for everything that for sequencing, and I was able to yield about 880 nanograms of material, which was plenty of material for at least 4 SMRT cells. Okay, the second attempt produced much better sequencing results after standard consensus processing. So after standard consensus processing, we had 25 gigabases of HiFi yield, which for the maize genome is approximately 12-fold coverage per SMRT cell for a total of 24x coverage. If you're interested in seeing this data, it's been published along with 4 other HiFi data sets, which include mouse, strawberry, mountain yellow legged frog, and a mock metagenomic sample. So definitely encourage those of you interested in analyzing HiFi reads or in developing tools to download any of these, any of these data sets. Okay, first, one of the most frequent questions we were asked is, can I multiplex HiFi libraries together for Genova Assembly? And the answer is yes, but only for genome sizes below 600 megabases. So this is great for organisms with smaller genomes, and it may even be possible to increase the plex level with even smaller genomes. So one example of this is a project we did multiplexing two individual mosquitoes. So DNA was extracted with a modified QIAGEN mag-extract protocol, and the 230 nanograms of resulting high molecular weight DNA for each individual was brought into library prep and then pooled in equal molarity. The cell was sequenced with the standard 30-hour collection, and from that we were able to get roughly, I think, a little over 6 gigabases of HiFi data per individual, which for mosquito equated to approximately 25-fold coverage per individual. Another frequent question we get asked is, what if I have a small organism but a large genome? So in other words, I have a limited amount of DNA, but I have a large genome that would require more than one SMRT cell. What do I do? So one answer to this is using a combination of the low and ultra low DNA input protocols. So the ultra low input data helps improve overall coverage, while the low input data helps cover those high and low GC content that may have been biased in the PCR amplification from the ultra low protocol. So here we're showing real world data from a single female tick, where we did 2 cells of from the low input DNA protocol and 2 cells from the ultra low input protocol. So this data was combined to produce a high quality assembly, as we're showing here, using HiFi ASM and purge dupes. And the total assembly length ended up being 2.38 gigabases, which agrees nicely with the expected genome size of 2.3. If you're curious, what I'm not showing here is that the total HiFi yield for the low input fraction was 40 gigabases, or about 20 gigabases per SMRT cell. And the total HiFi yield for the ultra low fraction was 53 gigabases, so approximately 25 gigabases per SMRT cell. What do I do if my sample is already degraded? So I get this asked pretty often. So it's not great if you're doing a de novo assembly project, but if you're dealing with a metagenomic sample, not all is lost. So for example, with a metagenomic sample, the fragment length is not as critical as it is for a reference sample. So therefore you can proceed. So shown here is an example of 10 stool samples extracted using the QIAamp parafecal DNA kit. The important aspect of this is that that I want to highlight is the differences in sizes between the tape station and femtopulse systems. The collaborators submitting these samples used the tape station to QC the samples. When I received them, I measured the size on the femtopulse, which showed that the samples were more degraded than previously thought. This can shift the expectations of a project. And so, in addition, it can also affect the size selection decisions you make downstream, and it will affect the molarity calculations you make when loading the instrument. So it's real important to use the appropriate technology for measuring DNA size so you get an accurate picture of the material you're dealing with. Because if you're using something like the tape station, just always know that it's going to overestimate your DNA size. So expect that the true size is probably several kilobases smaller than what the tape station is estimating for you. So despite being degraded, these samples are rather pure, so they sequenced just fine. So out of those 10 samples, 4 pools were made using 4 libraries per pool. And each of those pools were sequenced on a single 8M SMRT cell in the Sequel II system. So, and I did the size selection step using the diluted ampere PPBs to remove the fragments less than 3 kb. So you can see here the base yields were good. The P1 percentage was rather high and good, and the read lengths were good. So this all yielded nice nice HiFi data for the metagenomic project. Okay, so we also often get asked what size selection method is the best to use for HiFi sequencing. And the answer is going to depend on what application is being run. So if you're doing variant detection project with somewhat low coverage, it might be better to use the Sage ELF system in order to get a narrow slice of DNA fragments. What this will help with is making all those HiFi reads that you get from that data roughly the same, to have roughly the same quality score. So all the data in that will be around Q30 or Q32 or whatever it is, what size you're working on. So for Genova Assembly projects, we don't see much of a difference between the Sage ELF system or the Blue Pippin systems. The larger tail of the Blue Pippin system, though, may help bring longer HiFi reads into the assembly, which can be very beneficial. But it could lead to, because you have a lot of high weight fragments there, like say greater than 30 kb, it could lead to decreased HiFi yield overall. But for the most part, it's sort of this trade-off, right? Because some of those larger fragments may not get enough passes with the polymerase. But with enough coverage, the mean quality score is not too much of a concern. If it is a concern for you, and you want to try to figure out ways to improve that median HiFi read quality score, and you are using the Blue Pippin, you can try to employ this double shearing method that we've recently been using to decrease those fragments that are greater than 30 kb and compress the fragment size distribution down. So this we tested this two-cycle shearing method on multiple samples for structural variant detection, and it works really well. And so as we show here, using the two-cycle shearing method can improve the median HiFi read quality from 29 to 31. It also has the advantage of leading to sort of more consistent loading and lower on-plate concentrations. Okay, and here's an example from the results of a patient DNA sample using the two-cycle shearing method. This is a single SMRT cell, and it yielded 2 million HiFi reads with a median quality of Q30 or 99.9% accuracy, with a mean HiFi read length of 17,212 base pairs. Yeah, so this is a really good data and a really good method to explore if you're interested in sort of compressing that size distribution and increasing that median HiFi quality score. Okay, so what if your samples clog in the hydropore or not shearing very well when you're using the Megaruptor system? So there's a few things you can try doing. You can heat the sample up at 37 degrees for 10 minutes, and then try to mix it with a wide bore tip. If that's not not helping the genomic DNA flow smoothly in that tube, then you could try just mixing it with a standard P200 tip until the DNA is flowing more smoothly. This will shear the DNA a little bit, but it's not going to shear it as much as, say, using the Megaruptor G-tube systems. So once you believe the DNA is sort of homogenized in solution, you can re-quantify it on the qubit system. But I recommend quantifying from 3 places within that tube, from the top, the middle, and the bottom. And then once you have that concentration reading, dilute the sample to 10 nanograms per microliter, or just dilute it to the lowest amount possible with the total amount of DNA you plan on shearing, given the volume you're going to use. So if you want to shear like 5 micrograms of material, you may need to use a slightly higher concentration here. And then once you're done shearing, then obviously QC it on the appropriate platform. Here we're highlighting the femtopulse, but just use whatever platform is appropriate for your lab. in your sample. Okay, so oftentimes, especially for new customers, we kind of ask, what do the metrics look like for a good HiFi sequencing run? Generally, the raw base yield should be, greater than 300 gigabases for a good run, and then hopefully you can see that polymerase read length around 80 kb if you're using, inserts, 15 kb or greater. You want that P1, the productivity metric, to be north of 50%. This is usually indicative of a good sample, good loading, and very few early terminations. And another kind of overlooked statistic is the local base rate. You want this to be somewhere between 1.8 and 2.1 base pairs per second. That indicates that the polymerase is proceeding smoothly, and there's no issues with DNA damage or a contaminant within the sample. Now, as I promised earlier, here's that first B73 sequencing run I did that used that Sage ELF 1040 kb protocol. Now, this would be an example of what a low yield HiFi run would look like. So here, you notice the base density plot at the bottom left. You have this really high peak on that left side near the y-axis. This is indicating that there's a lot of early terminating reads. And then that's going to translate to just a lower number of Q20 reads. And, that means, if you're seeing that, then likely, it's a result because of just a short read length here. So the mean read lengths for this sample were barely over 2 passes of the insert size we were actually sequencing. And another, statistic here, metric is the low average base rate. You see here was 1.4 and 1.57 respectively. So this all sort of adds up to sort of indicate that the DNA was damaged, and we had verified that doing some additional experiments that I'm not showing here. That damage was occurred during that size selection step. So if you're interested in reading more on evaluating run performance and run QC, you can download a copy of our guide shown here, created by our tech support group. And the link right there is on the slide. So, we pride ourselves in our customer support. So just reach out to your local FAS or our tech support group, and somebody can get back to you and help you. So yeah, don't be shy about asking questions and reaching out for help.
Okay, thanks.